Ever notice how some of the simplest things can be surprisingly complex? Like you pick up your phone, start scrolling, and bam, you're suddenly down a rabbit hole of code and algorithms.
It's fascinating, isn't it? How something so commonplace can be built on such intricate foundations.
Exactly. And today, we're diving into a world that's both familiar and full of microscopic mysteries. The world of bacteria.
Specifically, we're going to unlock the secrets of a clever little tool that helps scientists identify certain types of bacteria. The Phenylalanine Deaminase Test.
Phenylalanine Deaminase Test sounds kind of like a mouthful.
It is a bit of a tongue twister, but don't worry, we'll break it down.
Okay, good, because I was going to say, I need a translator here.
Well, think of it this way. Not all bacteria are created equal. Some are harmless, some are actually good for us, and then there are the ones that need a closer look, right?
Right, like a bacterial lineup.
Yeah.
We need to figure out who's behind the microbial mischief.
Precisely. And this test, it helps us do just that. It tells us whether a bacteria can break down a specific ingredient called phenylalanine.
Phenylalanine? That rings a bell. Isn't that an amino acid?
You got it. It's one of the building blocks of protein. And for some bacteria, it's a favorite snack.
So it's like setting out a buffet and seeing which bacteria go for the phenylalanine.
Exactly. And the Phenylalanine Deaminase Test is how we figure out who indulged. It all starts with a special petri dish called a phenylalanine agar slant.
Phenylalanine agar slant. Go on.
It's essentially a nutritious slope, kind of like a bacterial buffet, designed to see if our microscopic suspects have a taste for phenylalanine. We give them the food and watch what happens.
Okay, I'm with you so far. We've got our suspects, our buffet. But how do we know if they actually ate the phenylalanine? What's the giveaway?
That's where the real ingenuity comes in. It's all about a color-changing clue. You see, if our bacteria have been munching on the phenylalanine, they leave behind a byproduct, phenylpyruvic acid.
Phenylpyruvic acid. Catchy.
Right. And to reveal this calling card, we use a special solution called ferric chloride. It's like a detective's blacklight. It reacts with that phenylpyruvic acid and, drumroll please, that causes a color change.
Let me guess. It turns green.
You got it. A vibrant green means our bacteria are positive for phenylalanine deaminase, meaning they ate their phenylalanine veggies.
And if it doesn't change color...
Then we know they're not fans of phenylalanine, which tells us something important about their identity too.
Fascinating. So we're not just identifying bacteria here. We're getting a glimpse into their dietary preferences. But why does any of this matter? What's the big deal with phenylalanine and these color-changing bacteria?
Well, it turns out this ability to break down phenylalanine is particularly common in certain types of bacteria that, well, can cause some trouble.
Troublemakers, huh? Like who? Give me names.
Think Proteus, Providencia, Morganella. They're often linked to infections, especially in the urinary tract.
Okay, now those names are starting to sound familiar.
And believe me, when it comes to infections, you want to know who you're dealing with sooner rather than later. Yeah.
Absolutely. So we're not just talking about a theoretical science experiment here. This test has real-world implications for diagnosing and treating infections.
Exactly. And it all comes down to understanding a bacteria's appetite for phenylalanine. Who knew such a simple test could hold such important clues?
So we've got this bacterial buffet set up. We know who our usual suspects are. Walk me through how this test actually works in the lab.
Well, imagine this. We're in a lab, white coats on, and we've got our petri dish with that phenylalanine agar slant, our bacterial buffet, as you called it.
Okay, so far so good. We're plating our suspect bacteria on the phenylalanine agar. What then?
Then it's time to let the bacteria do their thing. We carefully place the inoculated slant into an incubator, kind of like a bacterial spa, nice and warm, perfect for them to grow, and, if they're up for it, to metabolize that phenylalanine.
So we just leave them to it for a while. How long are we talking?
We give them some time. Usually about 24 to 48 hours. Enough time for them to really dig in, And then comes the exciting part.
This is where you get to play detective, right?
Exactly. After the incubation period, we take our special solution, the ferric chloride, remember?
The black light, right. It reacts with, what was it again?
Phenylpyruvic acid. It's the byproduct those phenylalanine-loving bacteria produce. So we add a few drops of ferric chloride directly onto the slant.
The suspense is killing me. What happens?
If the slant turns green, that vibrant green we talked about, we know those bacteria have been busy breaking down phenylalanine. They're positive for phenylalanine deaminase. But
if the slant stays the same, kind of yellowish,
then we know our bacteria weren't interested in the phenylalanine. They don't possess the enzyme to break it down, which tells us they're likely a different kind of bacteria altogether.
So each test is like a clue, helping us narrow down the possibilities.
Exactly. And that's how we start to identify these microorganisms and understand what they're capable of.
And that knowledge, knowing what a bacteria can or can't break down, that's what helps doctors make decisions about treatment, right?
Absolutely. Especially with those infections we talked about, urinary tract infections.
Yeah.
Knowing whether you're dealing with a proteus, a bacteria that loves phenylalanine, or something else entirely, it can completely change the course of treatment.
It's amazing how a simple test, looking for a color change on a Petri dish, can have such a huge impact.
It really is. And it's a great example of how microbiology, a field we often can't even see, plays a critical role in our health and well-being.
It's really remarkable when you think about it. The seemingly simple test, looking for a color change, can have such a big impact.
It really highlights how connected science is to our lives. We think about microbiology, and it's this invisible world, but the impact is anything but invisible.
It's true. We can't see it, but it's there, working behind the scenes. Kind of reminds me of that saying, for want of a nail, except in this case, it's for want of knowing if a bacteria can eat phenylalanine.
Exactly. And that's what's so fascinating about this field. It's full of those aha moments.
Yeah.
It makes you realize how much we still don't know.
It really does. So what other kinds of tests are out there? I mean, if this is just one example, what other tools are scientists using to uncover the secrets of bacteria?
Oh, there are so many. And each one gives us a little piece of the puzzle, helps us understand these microorganisms even better.
And the more we learn, the better equipped we are to deal with the problems they can cause, right? Like you said, antibiotic resistance, new treatments.
Absolutely. And it goes beyond that even. We can learn how to use microbes to our advantage, develop new technologies, solve environmental problems. The possibilities are pretty amazing when you think about it.
It really drives home the point that science isn't just happening in a lab somewhere. It's all around us, impacting our lives in countless ways.
Exactly. So for our listeners out there who might be thinking, phenylalanine, what's that? It's about understanding the bigger picture.
Right. Even if you're not a scientist, these discoveries, these tiny details, they matter.
They do, because they all contribute to a healthier and safer future for all of us.
That's a great point to end on. So the next time you hear about some new scientific breakthrough, or maybe even a food safety alert, remember the Phenylalanine Deaminase Test.
And remember all those unseen microbes that have such a visible impact on our lives.
That's it for this deep dive into the fascinating world of bacteria. Thanks for listening.